In this video we will discuss growth curve. Growth curve is related to growth of bacteria, specifically. Bacteria are microscopic, they increase in population by make lots of copies of themselves. Unlike growing in size like us humans. Here conditions refer to the conditions under which bacteria are growing. Conditions here refer to the environment of bacteria in which it is growing. These include temperature, pH, oxygen, etc. If optimal conditions, meaning best conditions, are given, the bacteria grow exponentially. Under optimal conditions, bacteria can grow in large numbers in short periods of time. Now let's try to understand what is growth curve. When we talk about growth curve, we are dealing with population of bacteria. Rather than a single bacteria, keep in mind. If we have to define growth curve, we will say, growth curve is a graphical representation that shows how a population of organisms, like bacteria, increases over time under specific conditions. It shows different stages of growth bacteria go through as they multiply in the environment. So what are these stages or phases of growth? When a bacterial culture is grown in a closed system, a batch culture, the population typically undergoes 3 phases. These are 1. Lag phase, 2. Log phase, 3. Stationary phase, 4. Death phase. To obtain a growth phase, we have to grow bacteria in a culture medium. After bacteria are introduced into a new environment or culture medium, they enter the lag phase. So what happens in this phase? The bacteria are not dividing. But bacteria are metabolically active. They are synthesizing enzymes, proteins, RNA, and other molecules necessary for cell division and adapting to the new environment. How long a lag phase lasts depends on various factors. 1. The type of bacterial species. 2. Condition of the cells. 3. Richness of medium. There is little to no increase in the number of cells, but a lot of cellular activity is taking place to prepare for active growth. We call it a lag phase because during this phase, bacteria do not start dividing immediately after being introduced into a new environment. Instead, they lag behind in growth as they adapt to the new conditions, e.g. nutrients, pH, temperature, repair damaged cells, if needed, synthesize enzymes and proteins needed for division. Next is log phase, also called the exponential phase. During this phase, bacteria start dividing at a constant and rapid rate, leading to exponential growth. The rate of growth is determined by the generation time, the time it takes for the population to double. In the log phase of the bacterial growth curve, the rate of growth is very high because bacteria are multiplying rapidly. In other phases, the rate of growth may be slower or even negative, like in the death phase. So yes, as you can guess, there are lots of bacteria in this phase. What's the difference between rate of growth and generation time? We will look into it in detail. Rate of growth equals. The rate of growth refers to how quickly something increases in size or number over time. In the context of microbiology, the rate of growth typically describes how fast a population of bacteria is multiplying. Generation time equals is the amount of time it takes for a population of bacteria to double in number. It's like a measure of how quickly bacteria reproduce. This phase is characterized by the most active period of cell division. Duration. The log phase continues as long as the conditions remain optimal, including the availability of nutrients and space, and the absence of toxic waste products. Key point. This phase is where the bacterial population doubles at a consistent rate, leading to a steep rise in the number of cells. It's also the phase where the bacteria are most sensitive to antibiotics, since they target actively dividing cells. As resources, like nutrients and oxygen, become depleted, waste products accumulate, the growth rate slow. The number of new cells being produced equals the number of cells dying. The total population size remains relatively constant during this phase. If we talk about duration of the stationary phase, it can last for varying periods, depending on the environmental conditions and the bacterial species. Key point here is that although the number of viable cells remains constant, some bacteria may start producing secondary metabolites like antibiotics or spores during this phase as a survival strategy. Duration. The length of the death phase can vary. In some cases, a small number of cells may survive and enter a dormant state. Key point. The death phase illustrates the decline of the bacterial population, and the environment may become unfavorable for bacterial survival. So, the bacterial growth curve is like making popcorn. Lag phase. Kernels are heating up, bacteria are preparing. Log phase. Kernels are popping rapidly, bacteria are multiplying quickly. Stationary phase. Popping slows down, bacteria growth slows. Death phase. Kernels burn if left too long, bacteria die off. Working at a new place is like the lag phase of a growth curve. You're adjusting, observing, and trying to understand your surroundings. As you get more comfortable and start working efficiently, you move into the log, exponential, phase, where your performance rapidly improves. Once you master your tasks and settle into a routine, you reach the stationary phase, where growth levels off. If challenges or monotony set in without new opportunities, you may enter the decline phase, just like cells slow down when resources run low. This concept is foundational in microbiology, as it explains the dynamics of bacterial populations and informs many practical applications in medicine, industry, and research.